Good evening, everybody. It's my pleasure to welcome you this evening. My name is Chris Gonzalez. I'm a co-director of the Center for Genomics and Systems Biology here. And I have the microphone. Can you hear me? Yeah. Oh, better? Should I lean in like this? Okay. My name is Chris Gonzalez. I'll start over. I'm the co-director of the Center for Genomics and Systems Biology here at NYU Abu Dhabi and an affiliated faculty from the Department of Biology in New York. And this is a public lecture that we've held every year in association with our annual symposium, which is ongoing right now, and I hope that most of you are attending. We just finished the first day, and we have another day and a half, so please come join us if you haven't already. I'm going to introduce Mike Peruganen, who is the Dean of Science in New York and another co-investigator in our center here, who will give you a little bit about history about Nina Fedoroff.
Thank you, Chris. It's really a pleasure to be standing here in front of you to both welcome you to this public lecture and to introduce Nina Fedoroff. It's not saying too much by saying that Nina is really one of my scientific heroines. And if you know her background, she's known for many discoveries, but the one seminal discovery was she was the one who first molecularly isolated the transposable elements that Barbara McClintock first described in maize. In 1983, the year that Barbara McClintock won the Nobel Prize for her discovery of transposable elements. Nina, that isolation is really one of those classic molecular genetic experiments. It was published in Cell, and for those of you who are biologists in the audience, I would urge you to read the two papers back to back that describe the isolation and characterization of the maize transposable elements A, C, and DS. She's not only a heroine of mine, she's also my academic grandmother because what? No, she is, because my PhD advisor was a postdoc with Nina and was on the team that isolated the transposable elements in maize. And Nina's work in plants have really revolutionized our understanding of the plant genome. Her isolation of transposable elements really led to an explosion of studies of how transposable elements were important in plant genomes. And she, in many ways, was the heir to Barbara McClintock. She was the one who made Barbara McClintock's discoveries real in terms of the molecular basis for the actions that Barbara had seen at the genetic level. Nina got her PhD from Rockefeller University and has gone through several faculty positions, first at UCLA, then at the Carnegie Institution of Washington, where she had isolated the McClintock transposable elements, then to Penn State, where she was one of the senior distinguished professors before she went to Cows in Saudi Arabia. But actually, in between that, she was the senior scientific advisor to the Department of State, where she advised both Secretary Condoleezza Rice and Hillary Clinton on scientific and technology matters. She also has many honors to her name, the most prominent of which in 2006, I believe, she was given the National Medal of Science, which is the highest scientific award in the United States. She's, of course, a member of many academies, including the U.S. National Academy of Science. And after stepping down from Cows, she is still very active in the Washington, D.C. area. It's certainly a pleasure to have Nina here. I haven't seen her for a few years. Actually, I was at Cows when I last saw you, and I'm looking forward to your talk, Nina, and welcome to NYU Abu Dhabi.
What a nice introduction from a grandson. I have to tell you a secret. I'm already a great-grandmother. So I'm delighted to be here. It's really nice to be back in the Middle East, and I think we have a few Cousties joining us tonight as well, including Inas, who was my wonderful laboratory manager when I was at Cows. So on with business. We live in a mobile, technologically sophisticated, largely urban civilization. Our shops and our supermarkets are just loaded with fantastic produce. We have an incredible system that brings food from wherever it's grown in the world to those who can buy it. Most of us don't have a clue of what it takes to produce that food. So I'm going to start by telling you, just briefly, how we got to where we are today, which is a world of 7.5 billion human beings awash in food. Now, the population pundits are telling us that population growth probably won't stop until there are 10 billion or so of us, and we'll be richer. That means we have to produce roughly twice as much food as we grow today. The question is, how? So history first. For most of our evolutionary history, we were hunter-gatherers. We spent our days finding and capturing food. Then somewhere around 10 or 20,000 years ago, we began to shape plants and animals to our advantage and settle down to herd them and to grow them. That, of course, is called agriculture. And it allowed us to feed more than just our families. We could feed scribes and artisans and warriors and kings. What I'm saying is simply this. Human civilization emerged because we figured out how to grow more food than we consumed. Now, for millennia, civilizations rose and fell, generally lasting until the land wore out or until the neighbors invaded, having worn out their own. As plants extract nutrients from the soil, crop yields decline, making it harder and harder to produce enough food as the number of people grows. Now, a couple of centuries ago, the English cleric Thomas Malthus wrote that we were forever doomed to hunger and strife because our numbers invariably grew faster than our food supply. Now, when Malthus penned his gloomy thoughts and predictions, the global population was about 1 billion. So if Malthus thought the game was over when we were just 1 billion, how did we get to 7.5 billion today? Well, curiously, it was just about the time that he was penning his gloomy predictions that science began to enter agriculture in earnest. Now, over the next couple of centuries, innovations in several key areas, synthetic fertilizers, that's chemistry, genetics, and the internal combustion engine, transformed agriculture. Now, for those of you who are not plant biologists, plants are magic. They produce sugar out of water and thin air. Well, to be technically exact, it's the carbon dioxide in the air. But they also need nitrogen. And most plants can't use the atomic nitrogen that's so abundant in the air. Now, manure contains nitrogen in the right chemical forms, and it's been used as fertilizer since prehistoric times. But there isn't much much nitrogen in manure. Now, around the turn of the 20th century, Fritz Haber and Karl Bosch figured out how to convert atmospheric nitrogen to compounds plants can use. Today, that's done in huge factories around the world. We simply couldn't have gotten from 1 to 7.5 billion without synthetic nitrogen fertilizer. Genetics was critical as well. Nobel Prize laureate Norman Borlaug is considered the father of the Green Revolution that set the famine-plagued Asian countries on the road out of poverty. The Green Revolution was based on mutations that let crops take advantage of fertilizer, doubling and tripling yields. Mutations. Mutations are genetic modifications. Perhaps genetically modified organisms, GMOs, aren't as new as we think they are. Then there's the internal combustion engine. Agriculture was hard, physical work for most people for most of history. The populace was largely agrarian, even in the most advanced countries, until well into the 20th century. As machines gradually took over the work, fewer and fewer people could produce more and more food. Now, what happened to the people? People moved and continued to move into cities, which became hotbeds of invention and collaboration. Technology-based wealth generation accelerated, giving us all of the machines, gadgets, and comforts of modern life, even Twitter. In sum, our largely urban existence came about because we got better and better at growing food efficiently using science, chemistry, engineering, and biology, especially genetics. But before I get into the nuts and bolts of GMOs, which is rather controversial, let's glance at some of the future of our ability to feed ourselves, starting with our sheer numbers. Now, population growth is slowing, but it's not likely to stop much short of 10 billion. There are various slightly larger and slightly smaller estimates. Take your pick. Now, the important thing to appreciate is that as people get richer, they want to eat more meat. Switching from a largely grain-based diet, typical of less developed countries, to the meat-based diet of developed countries, ups the demand for feed grain, as much as tenfold for your favorite hamburger. Now, if we don't change what we're doing, now that, of course, ups the demand for land, but there isn't anymore. And if we don't change what we're doing, we'll be back to Malthus's predictions in our lifetime. Maybe not mine, but certainly yours and your kids. Then there's the warming climate. Our major food and feed crops were domesticated in a milder climate. Yields drop at higher temperature. Now, the X marks the spot of the very hot summer in Europe. It was just 3 degrees above the average for the last century. Now, we all heard about the 30 to 50,000 people who died. But what we didn't hear about is that crop yields decreased by 25% to 50%, depending on the crop. Now, that'll be an average summer in just decades, and a cool summer by the end of the century. Even if Donald Trump doesn't believe in global warming. And there's water, of course. Most productive agriculture is irrigated, and the most reliable water comes from deep underground, indeed from fossil aquifers. These are being exhausted ever faster everywhere in the world. All of these indicators suggest that we are indeed at last reaching the limits of the plant's land and water resources, yet the population is still growing. We're also becoming increasingly aware that we must produce food more sustainably than we've ever done before if we're to leave the land in reasonable condition for our children and grandchildren. So that means that we have to produce about 1.5 to twice as much food as we produce now, using less land, less water, less energy, less chemicals. While that seems like an overwhelming challenge, look at it this way. It was agriculture that powered the 7-fold expansion of the human population over the last 2 centuries. So all we need is just one more doubling of the food supply. How? Well, just as it was for agricultural advances, the ones that I've already described, the answer lies in science, largely a combination of biology and technology, and of course, some chemistry advances as well. Let me just give you a little glimpse of the agricultural technology of the future, and then I'll tackle the biology. Now, much of the technologies in the pipeline are already in use, albeit, if you look at it in a global context, underutilized as yet. Today's farm machinery is GPS guided and fully programmable. Now, plants absorb light in the visible part of the spectrum and reflect it in the near infrared. The graph shows the light that plants reflect. Healthy vegetation reflects more. Oops. Push the wrong button. So that's healthy vegetation. This is unhealthy or sparse vegetation. And this reflectance data can be used to map across fields, that's what's shown in this diagram. And this information can then be used to optimize everything from planting density to irrigation, fertilization, pesticide application, even to assess crop insurance needs, and throughout the growing season. Much of this can be done and is being done with drones, from diagnostics to agrochemical applications. And then there's the driverless GPS-guided harvesting machines. Indeed, the farm of the future, the large-scale farm of the future, might be almost completely run by machines. This is all possible. Imagine that. But high-tech farming faces both economic and societal barriers. It's expensive, so it only makes sense if it's done on a very large scale. But large-scale farming, often referred to pejoratively as factory farming, is generally regarded undesirable by city folks. Now, let me talk a bit about the problems that are particularly critical in this part of the world. It's particularly challenging because of both of the high temperatures and the limited supplies of fresh water. The past few decades, various forms of spray irrigation, and you see that still all around you, have been used very heavily. It's illustrated in this aerial photo from neighboring Dubai. But as groundwater is pumped out, seawater seeps in. And what comes out of the wells is increasingly salty. As well, evaporation from prolonged surface irrigation concentrates the solutes, evaporates off the water, inevitably building up salt at the surface until farming becomes virtually impossible. More than 20,000 farms in Abu Dhabi and this western region have been abandoned because of the increasing salinization of the groundwater. Now, driving across Abu Dhabi on a consulting trip a couple of years ago, I was amazed at the miles of abandoned fields full of nothing but irrigation tubing, the land's last crop. So what kind of technology is needed to produce food under these circumstances? Well, one approach is to explore the agricultural potential of plants that can grow in salt water. These are called halophytes, and there are a number of different ones, and they've evolved special mechanisms that allow them to grow in salt water. They either sequester the salt or they excrete the salt. Now, both approaches are being, exploited at the International Center for Biosaline Agriculture, which is in Dubai. Masdar and ICBA both have projects combining aquaculture with halophyte cultivation, small-scale projects. But it disappeared. Sorry. My view is that in the many discussions I've had about biosaline agriculture is I don't think that will ever replace the other kinds of agriculture that we're talking about, simply because it costs plants energy to get rid of the salt and that means that they grow rather slowly. There's not much domestication done, complicated genomes. And remember that we've been domesticating plants like corn and wheat and soybeans for 10, 20 millennia. So the most common approach is to move plants inside. And what you'll see throughout the Middle East is these kinds of simple tunnels or screen houses with a cooling wall at the end. That's what you see in the distance here. Simple cardboard, corrugated cardboard wall that fresh water is dripped over, and then a fan blows the water through to cool the greenhouse. So it's evaporative cooling. It's generally necessary because of the high temperatures in this part that are achieved in the summertime. In the wintertime, it's not so bad. But it doesn't save much water. It takes roughly the same amount of water to grow plants in open fields as it does to use dripping irrigation in an evaporatively cooled greenhouse. Now, here's a project that is happening just about 40 kilometers from here under the Abu Dhabi Food Control Authority. This is called the Bani Yas Center for Agricultural Research, and they just recently completed a fairly modern experimental greenhouse. What you see in this diagram is that they're comparing old-style tunnel greenhouses with more modern, much taller greenhouses, and within the taller greenhouses, there are two different types. There's a water cooled evaporative, a part of it that is cooled evaporatively in much the same way that the cooling is done in the tunnels, but at a much more sophisticated level. The greenhouses are much taller. There's more control of the incoming heat. And here's the high-tech greenhouse just coming online, and here it is fully growing tomatoes. Now, around the world, this kind of technology, which is used in most advanced greenhouses, produces on the order of 100 kilograms of tomatoes per square meter per year, using about an order of magnitude, 10% as much water as the simple tunnel that I showed you earlier. But there's still room for advancement in greenhouse design to reduce both water and energy footprints of food production here in the Middle East. This is a diagram illustrating the work of a graduate student who developed this concept of a closed greenhouse system working with me at KAUST. I think he's here, supposed to be here. Ryan? Not here yet. It turns out that he's in Abu Dhabi at the moment, and he was going to come over. He then carried, after I left KAUST, he continued to develop this, working with a membrane specialist, Dr. Susana Nunez, to produce the kinds of membranes that are required to carry out this kind of a concept. The idea is to use a liquid desiccant to cool the greenhouse by running the desiccant through a hollow fiber across whose membrane the water vapor passes into the salt solution inside of the fiber, carrying the heat with it. So that's the cooling part. Then that process is reversed. It's called membrane distillation. And you can separate the brine, which is then reconcentrated to be reused, and fresh water is regenerated and then reused in watering the plants. And of course, this can be driven by solar thermal energy. Now, here's what the rig looks like. This is the miniature greenhouse and all the monitoring equipment to ask whether the model actually works. And it does. Now the question is whether it can be scaled as the calculations suggest that it can. But it's only one of many ideas. It's not rocket science. This principle is actually used in air conditioning systems already. Now I'd like to go on to the biology. And GMOs, I think many people, how many are familiar with that terminology? Yeah, everybody knows what a GMO is. You'll see. Well, some see it as the way forward, even miracles for a hungry world. Others are convinced that GMOs are a public health hazard and a menace foisted on us by big biotech companies. Well, just Google GMOs, and you'll quickly see GMOs have been blamed for everything from farmer suicides in India to tumors in rats, in every manner of human ill, from autism to obesity to infertility and cancer. Parenthetically, the same kinds of indictments were made against the Green Revolution strains, particularly the one about fertility. Now, none of this is true, but there is a growing body of what can only be called, in contemporary terminology, fake science around GM food. This paper was retracted because the scientists used rats that spontaneously develop tumors as they age, so it's claimed that the tumors were created by his feeding the animals GM corn was simply bogus. But the paper popped up again in a less reputable journal, and that's a problem for science today. And the images of the rats with grotesque tumors are still circulating on the internet. So today, the man and the woman on the street generally doesn't know what a GMO is, but they know it's bad. Watch this.
Do you try to avoid GMOs in your diet?
I do. Yeah.
Tell me why.
There's just a vibration with GMOs. For me personally, just something that I don't particularly want to put into my body.
What does GMO stand for?
Genetically modified. Genetically modified.
Yo.
The. I don't know.
Do you try to avoid GMOs in your diet?
Yeah, absolutely.
Why is that?
Just the effects, I guess, on myself.
What does GMO stand for?
Oh, man, putting me on the under the grill. Let's see. I don't even remember.
Do you try to avoid GMOs in your diet?
Absolutely,
Why?
because they're not good for you.
What is a GMO?
It's genetically I don't know, what is it?
If you are eating whole foods, you want to eat what you're eating. You know what I mean? You want to eat what you see. And so, and you're avoiding processed food, but when the whole food is somehow contaminated, that's kind of making it a moot point.
What does GMO stand for?
Genetically manufactured. Oh.
What is a GMO?
General modified ingredient, right?
What is a GMO?
It's something modified.
Do you try to avoid GMOs?
Sometimes. Not a whole lot, but, I try.
What is a GMO?
I don't know. I swear. I was just going along with it. I don't know what GMO is.
What is a GMO?
I don't know. I know it's like some corn bad stuff, right? I know it's bad, but to be completely honest with you, I have no idea.
What is a GMO? It's an acronym for genetically modified organism. Today, that means an organism modified by molecular methods. But as I've tried to tell you, we've been genetically modifying organisms for millennia. Okay, this diagram illustrates the tools that created the first generation of GMOs. It's called recombinant DNA technology. Enzymes from bacteria are used to snip and piece together bits of DNA from different kinds of organisms. The parts represented in purple, in gray, from a bacterium or a virus. And the part represented in purple is a segment of DNA from another kind of organism, be it a plant, animal, or another microorganism. The hybrid is called the recombinant DNA molecule. The The pieces are sealed together with an enzyme appropriately called a ligase, then slipped into a bacterium and multiplied to great numbers, like millions and billions. That process is called cloning. This in turn makes it possible to completely understand the snippet of DNA by techniques that include DNA sequencing. The cloned gene can then be modified and expressed in a completely different organism. Now for plants, that means enlisting the help of nature's genetic engineer, Agra Bacteria. If you've ever visited Washington, D.C. at cherry blossom time, which is earlier and earlier every year, you might have noticed that the old cherry trees are covered with lumps like these. Such lumps are caused by Agrobacterium, a species of soil bacterium that has developed the trick of transferring DNA into wounded plant cells to cause them to grow into these tumors. They don't kill the plant, but incidentally, they produce food for the bacterium. That's shown diagrammatically here, and it's carried out by genes encoded by a mini chromosome called the TI plasmid, and the piece of DNA that's transferred is the tDNA. Now, scientists figured out how this bacterium does the transfer, and so what they do now is they take out the tumor-causing genes and they put in a gene of their choice, and then allow the bacterium to use everything it knows how to do and carry the piece of DNA into the plant cell where it's integrated into the plant's DNA, creating what's called a transgenic plant. I suspect you've heard that term. Or if it's a crop, it's called a genetically modified or GM crop or a GMO. Perhaps the best known GM crops are BT corn and cotton, which are protected from major pests by a gene from a bacterium called Bacillus thuringiensis. The crops are called BT after the bacterium, although the added gene goes by a different name. The bacterium itself, Bacillus thuringiensis, has long been used as a safe biological insecticide by everyone from home gardeners to organic farmers to conventional farmers. The gene that makes it an insecticide codes for a protein that is toxic to the insect pests that bore into the stalks, as you can see here, and the ears of corn and into the cotton boll, causing that much damage. Now again, to stress that particular protein is toxic to this subset of insects, but not to people or animals, because they don't have a receptor that interacts with it. Now, adding just that one gene makes the corn and cotton resistant to the pests. This is BT corn, this is BT cotton. It's important to appreciate what a step this is towards sustainability. Globally, we use about 1 billion pounds of chemical insecticides a year. These kill both pests and beneficial insects throughout the area sprayed. The GM plant affect only the pests themselves and only those that attack the crop plant. Now, GM animals and insects have had a hard time getting out of our rather arcane regulatory system. A little company called Aquabounty created a transgenic salmon that has an extra growth hormone gene that's under a promoter that is not regulated seasonally. So what that means in common lay terms is that the wild salmon only grows part of the time, but it eats all the time. Whereas this salmon grows all the time, so it's not a gigantic salmon. These are two salmon at the same stage of development, one of which has been growing continuously and one of which has not. So it grows to market size much faster and on less feed. That makes it economically feasible to set up aquaculture facilities, completely recirculating aquaculture facilities on land. And why is that important? Well, most of the fish, the salmon that you eat today, the wild salmon is overfished, and much of the salmon that we eat today is farmed. Farming has major problems because they're in open cages, so the food waste, the waste from the fish It pollutes the ocean. It's unused nutrients that then cause things like algal blooms. And then diseases and pests like sea lice are a huge problem for the aquaculture industry right now. Okay, now that's just an example. This thing has been stuck in what I call regulatory purgatory in the US for almost 2 decades. It was finally approved in 2015. It's identical to its wild counterpart. There is no nutritional or biochemical difference. But right now, there's a political hold on it by our senator from Alaska, who feels that this poses a threat to the wild-caught salmon industry. Okay, now there's just one other that I'd like to mention, and that's the Oxitec genetically modified mosquito. It was developed by a little company called Oxitec, now a subsidiary of a bigger company called Emerita. The Oxitec mosquito carries what amounts to a lethal gene. And what the company has done is put that lethal gene under a promoter that silences the gene while the animals are being raised. They sort the males from the females. They raise only males. Males don't bite. The males' single job is to go out and find wild females and mate with them. Now, it's very species specific. There's one domesticated species of mosquito, Aedes aegypti. That's the one that carries all the nasty viruses like dengue, chikungunya, West Nile virus, yellow fever virus, and now Zika. This has been under trial in a number of countries for almost 10 years already. It's been spectacularly successful in Brazil, where it knocked back, in the areas where it released it, knocked back dengue by about 90%. Okay, let's talk a little bit about the impacts of these crops and then about the safety. Now, GM crops have been adopted by farmers faster than any crops in the history of humanity. They've had a significant impact, both economically and environmentally, all good. Here are some factoids from 2016. GM crops were grown by roughly 18 million farmers in 26 countries on 457 million acres. Now, more than 90% of the farmers growing them are smallholder, resource-poor farmers in developing countries. The profits, overall profits, were roughly equally divided between the developed and the developing worlds. So it's not a simple case that this only benefits big farmers. Now, this simple diagram summarizes the economic impacts and the environmental impact. Pesticide use decreased by more than 1/3, and pesticide costs by almost 40% over the period from 1996 through 2014 when this study was completed. GM seeds cost more than conventional ones. This added cost is largely offset by savings in labor and chemicals. So production costs over this whole period increased by less than 4%. Yields increased by more than 20%, and farmer incomes by almost 70%. You can imagine this is a percentage, so that most prominently benefited farmers in developing countries. Okay, so is it safe to modify organisms using molecular methods? Scientists have been creating and studying organisms, that is microbes, plants, and animals, modified by molecular techniques for half a century now. They've been studied and studied and studied. The EU issued this report in 2010 in which it summarized 25 years of biosafety research costing upwards of 300 million euros. Main conclusion: crops modified by GM techniques are no more dangerous than crops modified by other methods. Let me be more precise. Crop modification by GM techniques is no more dangerous than crop modification by other methods. That's not saying that all GM crops forever and ever are going to be safe. It just means that there is nothing that has surfaced in all of this time that would suggest that using these techniques is singularly dangerous. Every credible scientific body that's looked at the evidence has come to the same conclusion, including the US National Academy of Sciences, which has issued several reports over the years, most recent one in 2016. It runs to 400 pages and reviews literally thousands of studies. Its conclusion is that today's GM crops are harmful to neither humans nor animals. No evidence of harm. Now, the latest refinement in GM technology is referred to as genome editing. Now, the first generation of GM technologies added genetic material, but it wasn't possible to control where it inserted into the genome. The real prize has always been figuring out how to change just the gene you want to change and just the way you want to change it. Now, the newest and hottest gene editing tool is called CRISPR-Cas. You probably have heard that term already. It's in the news quite often. This is a tool derived from a bacterial defense system comprising a nuclease and a sequence-specific RNA molecule that targets it with considerable precision to the gene of choice, whose sequence is represented in the RNA that the enzyme carries. Now, the enzyme itself is represented by the green blob in the background, and the guide RNA that tells it where to go is shown in blue. And of course, scientists can change that sequence to make it find just the gene they want to change. Once the complex is formed and the guide RNA takes the complex to the target site, the DNA is cleaved. And then repair processes that normally operate in the cell jams the two ends back together, usually trims a little bit, and so it creates little mutations of a kind, by the way, of exactly the same kind that nature produces. It's what all of our domestication is based on, same kind as the kinds of techniques that we used for crop improvement in the last century, chemical mutagenesis, radiation mutagenesis uses. So these changes, many of them will be indistinguishable from changes, genetic modifications produced by other methods, which is going to give the regulators is giving the regulators a lot of headaches. And if you really want to know what CRISPR stands for, it's at the bottom of this slide. And it has to do with how this system was originally discovered. Not important to know. Now, here are two examples of crops modified by CRISPR-Cas technology. Dr. Yang, a plant biologist at Penn State University, mutated a polyphenol oxidase gene in a common mushroom, reducing its tendency to brown when bruised or cut. You can see that purely white mushroom on the right. Now, perhaps even more impressively, a group of Chinese researchers developed a variety of wheat resistant to powdery mildew by the simultaneously silencing of not just one gene, but 3 different genes, because wheat, some of you came to the talks this morning, wheat has 3 different genomes. So there's a copy of this gene in each genome, and of course, there are 2 different copies, one from mama and one from papa. So there's 6 different genes, and they were able to silence all of them. This could simply not have been done by previous methods. I think that the possibilities of attacking plant diseases and pests with these tools, whether applied to plants, the pathogen or the vector, are truly limitless. Let me show you just one example. Development has been much in the news recently is a hornless dairy cow developed by a company called Recombinetics. There are breeds of beef cattle that don't have horns. There are no hornless dairy cattle. So calves are generally dehorned for safety a painful process for the calf since it's commonly done by burning off the developing horns. Now, recombinants used CRISPR-Cas technology to produce a genetically hornless dairy cow by knocking out exactly the same gene that is mutated in the hornless beef cattle. Now that, again, could not have been done by conventional breeding in a denumerable amount of time. Conventional breeding tosses all of the genes of both parents together, and of course, dairy cattle and beef cattle have been bred for many, many generations for very different purposes. Throw that all into the same pot and then figure out how to get rid of the traits you don't want and keep the traits you want by backcrossing. Now, all of this sounds very sensible. Physical dehorning is painful, genetic dehorning is painless. So why all the controversy about genetic modification? Well, I'm sorry to say that the answer is that the controversy, like many new technologies that are introduced into the marketplace, there are stories that circulated. We were all going to have brain tumors from cell phones or in microwave ovens, and all of that kind of goes away. We don't hear much about recombinant medicines. What would we do today without recombinant human insulin with the epidemic of diabetes worldwide? But in the case of food, it's a bit different. There are both individuals and organizations that promote and exploit GMO fears for profit. On the one hand, Greenpeace's anti-GMO campaign is one of its biggest money raisers. Why should they give it up? And then there's, in the US, the organic food industry, and in Europe, it has conducted a very deliberate campaign of vilifying GMOs to drive them out of the market. Their simple objective is to increase their market share. All laudable goals, but the impact of these vilification campaigns has been extraordinary. Around the world, years of careful GM work on important crops have been lost through destruction of trials and personal attacks on researchers. Golden rice, I think, is a particularly poignant example. This is a rice variety developed by Ingo Petrikis and Peter Beyer and their teams decades ago. They added genes that support biosynthesis of beta-carotene, a vitamin A precursor, in the rice grains, which don't normally express it. Beta-carotene is in the body of animals and humans converted to vitamin A, a critical vitamin. What you see here is the destruction of experimental fields in which golden rice is being grown in the field by demonstrators that were brought in. Sorry, there it goes, from nearby cities. They weren't local people, but there is such anti-GMO vehement and such antipathy that people will go and collaborate with the folks that want to tear this up and destroy it. And vitamin A, of course, deficiency continues to be responsible for more than 2 million preventable deaths annually, largely of women and children living in poverty. I've whetted your appetite for more information about GMOs, but I have to caution you, you'll find a mind-boggling amount of both misinformation, just plain vitriol as you Google your way into the subject. So I'm going to suggest that you start by viewing an extraordinary new documentary titled Food Evolution, produced by Scott Kennedy and narrated by Neil deGrasse Tyson, everybody's favorite scientist. You can find it on Hulu and iTunes. The New York Times said this about the film. With a soft tone and respectful to opponents, but insistent on the data, Food Evolution posits an inconvenient truth for organic boosters to swallow. In a world desperate for safe, sustainable food, GMOs may well be a force for good. And if you want more, as I hope you will, here are a couple of really solid websites. The first is the Genetic Literacy Project. and the other is academics' review, which digs into the scientific underpinnings of the more outrageous claims quite nicely. As its subtitle says, testing popular claims against peer-reviewed science. Now, I hope I've convinced you that the societal controversies and regulatory tangles around GMOs and whether we can resolve them have enormous implications for our ability to feed the 10 billion humans we expect to be clamoring for food in the not-too-distant future. The fact is that what people believe matters. Indeed, we humans are belief-based creatures, and beliefs don't change quickly or easily. We're surprisingly capable of doubling down on them when they're challenged. So I leave you with questions. Will we continue to ignore facts and cling to our fear-based beliefs about GMOs? Will we have the wisdom to invest in the scientific and technological advances, some of which I've tried to show you, that we'll need in order to provide everyone with enough to eat, even as the climate continues to warm and our numbers continue to grow? I hope I've shown you that we can. Will we? I don't know. Thank you. And I'm delighted to answer questions.
Yeah, and it's now open for questions.
And you notice they handed out vouchers for 10 dumb questions at the door, and if you run out, there's more vouchers. Yes. I think there are microphones running around.
Test. Coming from Canada, it seems like a little bit of warming would be kind of nice, and there's an awful lot of ground up there that currently isn't used for agriculture. That might be. There may be a place to get more ground.
For sure, but as much as it'll warm, you still have you're not going to change, unless you tip the planet, you're not going to change the length of the seasons. And of course, the seasons are longer the farther south you go. So don't know, how much the land that opens up can actually replace the land that is disappearing. I mean, you're not going to double and triple crop in Canada as you can in more tropical areas.
Yeah. Thank you so much for the talk. I had no idea about a lot of this, but it reminded me of vaccines and how, especially in the US, people are very, very suspicious of this. But the thing is, sure, there are anti-vaxxers in other places, but that's also very specific to the US. And I understand why a lot of people...
Oh, it's pretty widespread in England as well.
Okay. Ooh. So my theory, at least with anti-vaxxers, is that part of it comes because they don't trust the public health system in their country. And if you don't trust, for example, big pharma, or you don't trust those, then why are you going to trust this other side of medicine that might be okay? And I can see some correlations there in here as well, where GMO is often part of these large multinational corporations that humans don't trust. So I'm wondering, how do you switch the mentality of making sure that people understand what is a potentially sketchy company and what is the good in GMOs?
Yes. I think that the part of the issue there is that beliefs are often constructed out of pre-rational thinking about moral convictions. The business about being against Monsanto is largely about, oh, that's not fair. And many people think they control the food supply, where the reality is it's a company that if it doesn't make money, it goes out of business. So they have to provide value for the farmer. So, all of that stuff. But, once one of those beliefs is embedded, it's really hard to dislodge. We have lots of information about how beliefs form. We have essentially no research on how you change beliefs. People are in fact resistant to the idea of having their beliefs changed. So it's really a tough human issue. It's not about delivering more information, that's for sure. I'm sorry to answer with such, the thing about the anti-vaccination movement is, the heartbreaking thing is it's based on one paper that we now know was just plain fake science. The guy made up the statistics. That one paper that was then retracted, the guy lost his license to practice medicine, but it took hold so deeply that we now have outbreaks of diseases we didn't have before because people were vaccinated. I think that's turning around, certainly turning around in California, because school authorities are insisting that kids are vaccinated. I'm sorry, there was one down here, yes.
Giving it, and I'm sure that people will accept genetically modified food when they get hungry.
That is what, to my mind, makes food different from anybody can get sick. You may get better healthcare, worse healthcare, but you're not immune from disease. Yeah. If you have money, you can buy food. Okay? It takes an awful lot to push food prices to the point where the folks that make the decisions, who are largely city dwellers that work in offices, are hungry. It's such a small delta.
I accept everything you say, but I think you're being a little bit unfair on people's beliefs. I just drove here listening to a program on BBC World Service about the opioid addiction that is epidemic in America. And this has been driven by Big Pharma. We also have antibiotics that we've studied for many years. I'm a doctor. I've worked in places where I'm actually frightened to work because the resistance that we have. So people, when they have their beliefs, they doubt that scientists look entirely at the big picture. Now, you've presented a big picture, and I commend you on it. But we also know that people are hungry in this world, not for lack of food.
You're absolutely right.
We also know
today.
Yeah, that people that
half a century from now, probably not.
Yeah, but at the moment, there's not a lack of food.
Right. It's about poverty.
And there are other things that we can do. There is a disparity between the rich and poor, which is growing, although there's less and less people in poverty, I admit. But I think that there's a general suspicion of big business and the way in which it's behaved and the results of it, and that maybe scientists don't entirely look at the big picture. I'm sure that Robert Oppenheimer, when he was developing nuclear weapons...
As opposed to who is looking at the big picture? I mean, the consumer?
No, I mean, I agree, the consumer doesn't, but I think you're just being a little bit unfair on the beliefs of people. They're not always necessarily hard-held beliefs. They're actually rational conclusions that they've made based on the information that they have. If they've been here and watched some of your film, maybe they would know differently. But, it's very easy to laugh at people and say you don't understand.
Yeah, but hold it. I'm getting convicted of something that I shouldn't be being convicted of. I'm not hard on people's beliefs. I'm just acknowledging that beliefs are often formed from exactly what you say, what you have, what your friends think, what you read on the internet, which can be information, misinformation. Once solidified, they're pretty hard to change. And that, I would say, from data. So I have a research project on this with a social psychologist and a physicist at the Santa Fe Institute. And even our preliminary data suggests, because we're sampling people and the beliefs of the people around them. People that have a mixed group around them, some who are pro, some who are anti, are much more open, so the probability they'll change their belief system on information input is already clearly much higher than if all their friends believe the same thing. So, I mean, those are facts, not my assertions, all right? So, and I'm not trying to be hard, I'm just acknowledging that very often people point to scientists and say, well, you're just not communicating well enough. But communication is not just about delivering facts, which is often how it's interpreted. It's about connecting with people's framework, okay? Sometimes it's, you know, their religious belief system, sometimes it's just a moral belief system that is more secular. But the point is that those things are as important as rational evaluation of data one way or the other. Just how we work. Oh, okay. And there wasn't a question there, so hopefully there's a question here.
I'll try to ask a question. Thank you for your presentation. And my question is a little bit about this big picture issue. One of the things that I think surprised some people 100 years ago was recognizing that as countries got wealthier, fertility declined. I'm wondering if as countries get wealthier, meat consumption may actually decline. It's been going up so far, but I guess I'm wondering if it may, is it possible that it could decline? And this is part of the big picture because you are talking about doubling the supply of food in the world, but you put up a slide which has already indicated that overnight there could be a 10-fold increase in food if we stopped eating hamburgers. And so why is science the answer instead of a answer, which I think is sort of behind the question I heard before. I certainly think science is an answer.
It's the answer, but again, it's pretty hard to legislate what people eat. There's nobody in the world. You can be persuaded by information from health authorities, and I think worldwide you're getting good information from health authorities, but there's nobody that's going to force you. In fact, in my country, there have been efforts to put an extra tax on things that are bad for you, like sugary drinks, and people won't stand for it. So it's a really tough one. Yes, we should be. And the evidence is that there are parts of society that are gradually changing those habits, decreasing food consumption, but it's not a big fraction of the world population. Hopefully it'll increase as people understand more. I don't know.
I have a question that's rather specific about GMOs. And my feeling on this has changed slightly, and that is on labeling. So there's efforts in some states to label foods as GMO.
No, there's actually a national law now.
Okay, initially, of course, we had been fighting against this, but I'm wondering to what extent it might not be a bad idea to accept GMO labeling only because I suspect that it would also show a price differential. It might show a price differential between GMO crops and consumers can then understand some of the benefits. I know Mark Linus is also an activist.
He has, and I'm agnostic on it. I am keenly aware that the organic food marketers did this very deliberately in an effort to vilify GMOs. And I find that totally outrageous. I mean, there are some things that are just morally wrong in getting greater market share. And that's one of them in my book. Giving in, it's a tough one for me to swallow simply because traditionally, I mean, the Food and Drug Administration has been very clear. Unless there is a health or environmental impact, it doesn't require labeling. You know, and if the food is exactly the same nutritionally, you can label it because it's a GMO, and then so how do you prevent people from seeing it as a kiss of death, which is the way it was intended? And so far, they're succeeding.
One last question.
So I'm curious about the sustainability of increasing another twofold the world food production in terms of how much can the land give in terms of nitrogen, the carbon cycles, and so on and so forth. Has there been, projection modeling done on that, whether it at all can produce that much?
I think there's plenty of modeling about it. I think there's no doubt that it can, and it can do it much more sustainably than we do it now. I mean, just combining, using herbicides and genetically modified crops, you can cut down on the amount of soil tillage, and keep all the waste in the soil. And in much of the world, the yields, much of Africa, which has a good deal of fertile land, the yields are just a fraction of what they are in the most developed, highly productive countries. So I think there's no question that it can be. But, again, there are social barriers in there, because even the big humanitarian foundations like the Gates Foundation, they want to focus on the small farmer. And if you look at the history of developing countries, it's very similar to ours. It's the increasing efficiency of large-scale farming. It's the large-scale farming that feeds most of the world. But people are reluctant to think about that and reluctant to deal with the consequences, because when you displace parts of the population, you can't just throw them off the land. There have to be opportunities, other opportunities. And nobody's gotten that equation. Everybody looks at a little piece of it. And there's a reception afterwards, so you can follow me with questions until I collapse in a heap.
So hi, I have a question. What do you think about the herbicide-resistant GMO that was developed by Monsanto, and what will be the effect of an increase of use of this herbicide on the environment?
Well, the fact that it's being overused is a consequence of the fact that it's more benign than any of the other herbicides that have been developed. What's happening now is because there's this whole worldwide anti-glyphosate campaign. By the way, there's an agency of the UN labeled it without any experimental evidence as a probable carcinogen. That became a carcinogen, and yet there's, the evidence underlying that is very small. The problem with glyphosate is that it is more benign and it has been overused, and it's a form of selection. If you use the same herbicide year after year after year, you're selecting for resistant weeds. Now, what's happening now is that there are crops in herbicide combinations using older herbicides, and they've got their problems. If you're familiar with it, there's a whole brouhaha over dicamba, which has a volatile form and a not volatile form, and Monsanto, in its infinite wisdom, released the dicamba-resistant seeds before they released the low volatility dicamba. So the drift of the conventional dicamba has really brought a lot of lawsuits on their heads. So the point is that Monsanto actually went out of, in the big agrobiotech companies, or agricultural companies, agrochemical companies is the word I wanted, kind of went out of the business of developing new herbicides. And that, like we have a problem with developing new drugs. We have a lot of drug resistance, but there's not much incentive to develop new drugs. And the same is true with herbicides. What you need to prevent that kind of natural selection or artificial selection is you need a rotation of probably 3 different herbicides in the 3 corresponding resistant crops. 3 years is enough so that you don't build up the resistance. And so that's the real problem. The glyphosate stuff is largely it's a controversy without a basis.
With that, let's thank Dr. Anita Fedoroff. And